Hello and welcome to the Digital Plant Health video channel. So this is our first video here, and in this video I'll talk about smarter cultivar mixtures to improve plant disease management. So this is a scientific presentation that I'm going to give at the ICPP 2023 conference that will take place in August 2023 in Lyon, France. And this is in fact the biggest conference on plant pathology, the International Congress of Plant Pathology, and I'm recording this video for the benefit of those who cannot join the conference in person. But even if you do attend my talk at this conference, I encourage you to watch this video because here I have a little bit more time and I can cover some of the aspects of this research in somewhat more detail. So here we're trying to control Septoria tritici blotch, which is one of the most damaging fungal diseases of wheat, caused by the fungal pathogen Zymoseptoria tritici. And in this photograph, you can see some of the typical symptoms of the disease, those yellow to brown lesions. And also we can see those black round structures within lesions. These are pycnidia, the fungal fruiting bodies, where the pathogen forms its asexual pycnidia spores. Now, before I go on, I would like to thank all the people involved in this research. So I think it was a very fruitful collaboration between the team at the ETH Zurich, where I have been a group leader previously, Patrick Arista, Bruce McDonald, and Sebastian Bonhoeffer, and the team from Tunisia, led by Sarah Ben Barek and Amor Yayawi, who conducted a very interesting field experiment on cultivar mixtures. And so they represent the CRP wheat Septoria precision phenotyping platform in Tunisia. I'm also extremely grateful to all the sources of funding who made this research possible. So we are dealing here with a pathogen that has extremely large populations. So we have recently estimated that the number of Zymoseptoria tritici spores produced in a single hectare of wheat in a moderate epidemic year is several trillion pycnidia spores. And these are produced by several million unique pathogen genotypes. And these estimates we have obtained based on many years of field data and also decades of population genetic studies of this pathogen. So I think these are quite robust estimates. And they indicate indeed that this pathogen has remarkably large and remarkably diverse populations. And this is one of the reasons why it is so difficult to control the disease, because if we apply fungicides or we deploy disease-resistant cultivars, the pathogen populations will adapt. Fungicide-resistant strains will emerge, or virulent strains will emerge within the pathogen population and lead to the loss of efficacy of these control measures, which is accompanied by immense economic losses in wheat production. So we really need to rethink the way we apply control measures to make it more sustainable. So not only do we need, in the spirit of the integrated management of diseases and pests, we need to combine several different approaches to control the disease. We need to combine fungicides and disease-resistant cultivars, but we also need to introduce more genetic diversity into our crops using cultivar mixtures, and this is the focus of my talk here. But also, for example, using intercropping. And we also need to think about crop microbiomes, how we can adjust them to protect crop plants from disease. And we need to combine all these different approaches, not just at random, but in an optimized manner across space and over time. And for this, we need to conduct a lot of field experiments, but they need to be boosted by novel phenotyping technologies and also combined with mathematical modeling so that we can have a quantitative prediction of what can happen in the field. And so by combining all these different methodologies and all these different control measures, we can impede the pathogen adaptation and achieve evolution-smart crop protection. And this clearly will contribute to development of more sustainable cropping systems. And so in this talk, I will focus on how we can optimize cultivar mixtures using a combination of field experimentation, novel phenotyping, and mathematical modeling. So let's start with the field experiments. So in this study, we reported a field experiment that has been conducted on durum wheat in Tunisia using cultivar mixtures. So in this experiment, it was conducted here at the Beja field experimental station in Tunisia over 2 consecutive years with 3 to 4 replicates. And here we use this novel phenotyping approach where we collected large numbers of diseased leaves in the field, scanned them with high resolution, and used the computer program to determine the boundaries of lesions, but also to count pycnidia within lesions. So we had 2 key measures of disease severity: the percentage of the lesion covered, of the leaf area covered by lesions, PLACL, and also the numbers of pycnidia in each of the leaves, in each of the diseased leaves, which represents the capacity of the pathogen to reproduce within the host. Now we had used 3 different cultivars in this experiment: Karim, the susceptible cultivar that is widely grown in Tunisia, and then the 2 resistant cultivars, Salim and Monastir. And so here we firstly looked at how the disease severity, measured as the percentage of the leaf area covered by lesions, depends on the proportion of the 2 cultivars in the mixtures. In this case, we were mixing Karim, the susceptible cultivar, and the Salim, the resistant cultivar. And clearly, the pure stand of the susceptible cultivar has higher severity, and the pure stand of the resistant cultivar has lower severity, as we expected. But what is quite interesting is what happens in between when we have intermediate mixing proportions. So it turns out that adding just 25% of the resistant cultivar to the susceptible pure stand suppresses the disease down to the level of the resistant pure stand, and even a bit lower, as you can see here. This is actually very good news for the durability of such a mixture, because if you have just 25% of the resistant cultivar, this will impose much less selection on the pathogen population to adapt to this resistant cultivar as compared to the pure stand of the resistant cultivar. And so this mixture is expected to last much longer in the face of pathogen adaptation than the resistant pure stand. But what is quite interesting is that this effect was cultivar specific. When we mixed Karim, the susceptible cultivar, with a different resistant cultivar, Monastir, we observed something quite different. So here we have a gradual decrease in disease severity as we increase the proportion of the resistant cultivar in the mixture. So why is this the case? It is quite puzzling. And so for a long time we didn't understand the underlying mechanism, but then we used the theory of cultivar mixtures that we have developed previously to try to make sense of these findings. So this theory is essentially based on epidemiological modeling, using this type of susceptible infected compartmental modeling approach. And so just to introduce the simple variant of this model, so we have 2 compartments here. The first compartment is the H, the susceptible leaf area index, and the second compartment is I, the infected leaf area index. And so the model essentially consists of the system of 2 coupled differential equations. But essentially, just to put it in simple words, it is a mathematical way to describe how these 2 quantities, our state variables H and I, change over time. And so we consider here, for H, for example, we consider here a very short interval of time dt, and look at how much H will change during this small time. And the amount of this change is represented by dH. And so dH over dt is the rate of change of H. And here in the right-hand side, we can write down the mathematical expression of how we think this rate of change should depend on the parameters of the model and the state variables H and I. The right-hand side here consists of 2 terms. Firstly, we have the host growth. This is a positive term, so the amount of susceptible hosts increases because of the host growth, and so this term essentially describes a linear growth that is limited by a certain carrying capacity K. And the second term describes the infection process, and it is a negative term because the amount of susceptible hosts decreases due to infection. And so the rate of this decrease is proportional to how many infected hosts we already have, because these serve as sources of infectious spores. But it is also proportional to how many susceptible hosts we have, because if there are no susceptible hosts, there would be nothing to infect. And the coefficient beta here is the transmission rate. And we have the exact same term as the first term here in the equation for I, for the rate of, so this is represents the rate of increase of I due to infection, because so here this term is positive. But then the infected host cannot stay infectious forever. They lose their infectiousness at a rate mu times I. And so 1 over mu represents the average infectious period of the pathogen. Now, I would like to emphasize this transmission rate because from the perspective of the pathogen, the transmission rate beta describes or quantifies how fast the susceptible hosts H are converted into the infected hosts I. But importantly, from the perspective of the host plant, the transmission rate quantifies the degree of resistance of the host plant. So resistant plants can suppress the transmission rate and in this way suppress the epidemic development in the field. So it is quite useful to visualize this model in a flow diagram. So in this diagram we have boxes for each of the two compartments, H susceptible hosts and I infected hosts, and we have arrows that describe all the possible processes that we model in the system, the host growth, infection, and the loss of infectiousness. This model has a number of assumptions, so it is spatially implicit. We consider no stochasticity, and these are fairly standard assumptions in this type of modeling. But we also neglect the latent period, which is a surprisingly good approximation. As it turns out, unless your research questions specifically address latent period. Now, we also consider endemic equilibrium. It is not an inherent assumption of the model, but we do this just for simplicity. So essentially, after an initial dynamic phase, the two state variables H and I reach a sort of a stable equilibrium state. And so the assumption is that this state, the endemic equilibrium, is reached before the end of the growing season, or at least approached before the end of the growing season. Now, in order to model cultivar mixtures, we actually have to extend this model because we need to have at least two different types of hosts that would describe the two different cultivars used in the mixture. And so here we now have the second host compartment. So we have H1 as the compartment for the cultivar 1 and H2 the compartment for the cultivar 2. Otherwise, the structure of the model is exactly the same. Both of these susceptible hosts of the two cultivars can be converted into the infected hosts, but the transmission rates can be different, and this can represent different degrees of resistance of the two cultivars. And so this model corresponds to the system of three coupled differential equations with exactly the same structure, but just extended to the case of two different cultivars. And so how do we learn something from this model? Essentially, we need to solve it. And by solving it, I mean that we need to find how the three quantities H1, H2, and I depend on time. But in order to do that, we need to know what values they had initially. So we need to determine the initial conditions. And so if you tell me the initial values of the three functions, of the three quantities, let's say at the very beginning of the growing season, and also tell me all the numerical values of all the parameters of the model, I can then use a computer program to solve the system numerically or analytically and find out how the three quantities change over time. And then I can compute disease severity as a fraction of the infected leaf area index and the total leaf area index. And here we can consider disease severity at large times towards the end of the growing season, when it approaches endemic equilibrium. And this is quite useful because then we can compare disease severity predicted by the model and the disease severity measured in the experiment. But now let's still stay in the theory. So for a bit, so how does disease severity in the simple model depend on the proportion of the two cultivars in the mixture? So we have beta 1 larger than beta 2, meaning that the cultivar H1 is more susceptible than the cultivar H2. So H1 susceptible, H2 resistant. And in this case, we have quite a simple pattern. So disease severity simply decreases in a linear manner as we increase the proportion of the resistant cultivar in the mixture. And so here there is no real advantage of using a mixture. If you want to suppress the disease as much as you can, you are better off just planting a resistant pure stand. Now, but we can also consider a more interesting scenario where we have two distinct pathogen subpopulations, I1 and I2, and they can specialize, each of them can specialize on one of the host cultivars, H1 and H2. And so this model can describe this scenario when we can have cultivar specialization. But this, of course, depends on the values of the, now we have four possible transmission pathways. So whether we have specialization will depend on the values of these four transmission rates. And these can be arranged into this transmission matrix. And we can conventionally say that we have cultivar specialization when the diagonal elements of this matrix, beta 1, 1 and beta 2, 2, are higher than the non-diagonal elements, beta 1, 2 and beta 2, 1. And this reflects a simple fact that transmission on your preferred host cultivar is higher than the transmission on a non-preferred host cultivar. Now, if this condition is fulfilled, if we do have cultivar specialization, then disease severity exhibits quite an interesting non-trivial pattern as a function of the proportion of the two cultivars in the mixture. So it essentially decreases as we increase the proportion of the more resistant cultivar, but then the disease severity is minimized across a large range of values of intermediate mixing proportions, and then it increases again. And so in this case, there is a clear advantage of using mixtures, of using an intermediate proportion of the two cultivars in the mixture. And so by comparing these two curves, we can see that the mixtures can have a clear advantage in the presence of the cultivar specialization. So we can now look back at our empirical data, so for the case of Karim Salim mixture, and we can view this data set in the light of this theory. So we can actually fit the model disease severity to the data, and we can try to fit the model with the two hosts and one pathogen, to H1P, that describes uniform, that has three compartments and describes uniform pathogen population with no cultivar specialization. And we see that, well, it tries to fit it, but actually the fit is not very good. Now, if we try to fit a somewhat more complex model that we also just looked at, that considers two distinct pathogen subpopulations with four compartments, and includes the case of cultivar specialization. In this case, the fit is much better. So the blue curve fits the data much better, and this visual impression is also confirmed by the ecological information criterion, which is substantially smaller for this 2H2P model as compared to the 2H1P model. And so this is our preferred model in this case. But now, if we look at the second mixture that we can see, investigated in this field experiment, here both of the models converge to this case of the 2H1P model that does not have the cultivar specialization. So essentially, this is just a uniform pathogen population, and it results in a simple gradual, simple linear decrease of disease severity as we increase the proportion of the resistant cultivar in the mixture. And so this is our preferred model in this case. So the key take-home message of this talk is that cultivar mixtures have an advantage when the pathogen population exhibits cultivar specialization. And so thanks to this data model comparison, we can now hypothesize that in the Karim-Salim mixture, the pathogen population exhibits cultivar specialization. And this is the reason why the disease severity is suppressed across a range of intermediate values of the proportions of the two cultivars in the mixture. Now, in contrast to that, we can hypothesize that in the Karim monastery mixture, the pathogen population does not have cultivar specialization, and this is the underlying reason why we observe this simple gradual decrease in the disease severity as we increase the proportion of the resistant cultivar in the mixture. Now, this is still a hypothesis because there can be a number of alternative explanations of these patterns in the data. But this data model comparison allows us to formulate several predictions that can be tested in future experiments. So firstly, as I already mentioned, we hypothesize or we predict that pathogen population will exhibit cultivar specialization for Karim-Salim mixture, but not for Karim monastery mixture. And this can be tested in future field experiments where we can investigate the genetic composition of the pathogen population and see indeed whether it exhibits, whether there are strains that are preferentially that transmit better on the resistant cultivar, on the one of the cultivars as compared to the other cultivars, and vice versa. Now, the theory also predicts that in the case of Karim-Salim mixture with cultivar specialization, the pathogen population will be more diverse across these intermediate mixing proportion, because this is where we expect to have a coexistence of the two distinct pathogens populations that are specializing on the two different cultivars. And thirdly, these patterns are only possible if the genetic basis of resistance in cultivar Salim is different from cultivar monastery. And we can eventually test this prediction when we learn more about genetic basis of resistance in these two cultivars. This is all. I would like to thank you very much for your attention. And if you would like to ask a question or leave a comment, you can do so below the video, or you can just send me an email.